Uh, my name is Donna Rekovich. It's such an honor to welcome the Michael J. Fox Foundation here in Detroit for our third annual research salon here in Detroit. So thank you guys for coming. The Michael J. Fox Foundation for Parkinson's Research is a tireless group of individuals who are driving research forward in order to transform patients' lives and ultimately find a cure for Parkinson's disease and go out of business. Team Fox is the Foundation's grassroots community fundraising program with thousands of members worldwide who host unique fundraisers like ours, our fall gala, our golf outing, raffles, and other community fundraisers to support research. Sorry. Last year, thanks to all of you here and our supporters in the Metro Detroit area, Team Fox and the D was one of the top 4 fundraising teams in the world who raised over $250,000. Since inception, in 2016, Team Fox and the D has raised a total of $568,931, and 100% of that went directly to support the Foundation's high-impact research programs. It is the research programs that give us hope. Only through funding research can we have hope for a cure. When people say to me Parkinson's is incurable, I remind them of the diseases that were once incurable but have been cured in the last century, like smallpox, diphtheria, polio, measles, malaria, and even AIDS has been stopped. Through research, we can be sure that one day Parkinson's will be on the list of these once incurable diseases. I'm hoping, that will be cured in this century. When I was diagnosed with early onset Parkinson's in 2012, I wanted to take action and take control of my disease instead of letting the disease control me. That's why I enrolled in the PPMI study, a biomarker initiative through the Michael J. Fox Foundation. This study collects data from all over 1,600 Parkinson's patients and makes it publicly available for any researcher to access in order to figure out what's the target. The progress the Foundation has made in such a short history is inspiring, and we are so pleased to welcome a few of their members today to speak about the work that they do. Now it's a great pleasure to turn it over the microphone to Lizzie Holland, Advancement Officer at the Foundation.
Thank you so much, Donna, and the entire Team Fox Detroit committee who helped us organize this wonderful event. I'm thrilled to be here. As Advancement Officer at the Michael J. Fox Foundation, I have the pleasure of traveling throughout the country to meet face-to-face with members of the Parkinson's disease community and to speak at events like this. Many of you, including Team Fox Detroit, are very familiar with the Foundation's work and have supported our mission for several years. For those of you who are newer to MJFF, I thought I'd begin with a brief introduction. The Michael J. Fox Foundation launched in 2000 with a tremendous sense of urgency infused by having a patient as our founder. The one goal was to cure Parkinson's disease. As a patient-centered organization, we have an openness to high-risk, high-reward ideas. We take strategic risks to broaden knowledge of Parkinson's disease and produce tangible improvements in patients' lives. We do whatever it takes to get that aha moment so that we can move more drugs through the pipeline and to the pharmacy shelf. To date, we've funded over $800 million in research, over a third of which is led by researchers outside of the United States. We don't care where the most promising research is, we will just fund it. Since inception, we spend an average of 88 cents of every single dollar donated to the Foundation on research. In 2019, we plan to put another $100 million toward our robust research programs. Today's conversation is on the evolving Parkinson's research landscape, as well as the work that is happening at the Foundation. Before we begin, I have a couple of housekeeping items. I want to mention that every table has both feedback forms and note cards. Please use the note cards to jot down any questions you may have throughout the presentation and save them for our question and answer portion. You can raise your hand with your card throughout, and we'll collect them throughout the program. We also greatly appreciate any feedback you can provide to help us make the most of these events. Please turn in the completed feedback forms to the registration table after the presentation or hand them to any staff members. Now I'd like to introduce my colleague, Dr. Kuldip Dave, MJFF's Director of Research Programs. Kuldip leads the Foundation's priority interest in alpha-synuclein, an important protein linked genetically and pathologically to Parkinson's disease. He also oversees the emerging targets portfolio to identify and validate novel targets for PD.
Hi everyone. Can you hear me?
Yes.
Okay. First of all, thank you to Donna and Mike and the Team Fox Detroit family for inviting me again. I must have done something right last year, so I got invited again, and truly making me feel like part of your family, and I really appreciate that. So I'm Kuldip Dave. I'm a scientist at the Fox Foundation, and what I thought I would do today is give you an understanding of what our landscape looks like. You all want to know what therapeutics are in the clinic, what are we testing, and so what I thought I would do is go through a brief introduction on Fox Foundation and on Parkinson's disease, and then talk about some of the genetic targets and why genetics is so important to understand Parkinson's in general, and then go into all the different programs that are in the clinic, because I think it's important to set that up before I talk about the list of drugs that are currently being tested in Parkinson's patients. So I'll get started first with, and Lizzie talked about this, so we are the largest nonprofit funder of Parkinson's research, and we have a single mission, which is to find a cure for Parkinson's. And we don't keep an endowment, which means each year all of the money that's raised, our research bank balance goes down to zero at the end of the year, because we want to get that money quickly into the hands of researchers. We don't want to be sitting on that money and build an endowment. We want to be out of the job, and so the point is to not have a wealth of money to get it into the hands of researchers. Lizzie talked about this, but just a real quick thing, about $900 million in research programs funded to date, and if we do fund another $100 million this year, we would have hit the $1 billion mark. And I think it's a testament to you all in supporting us, raising money, and raising awareness for this disease that we're able to do this. So I'm gonna maybe start from here and work my way to the left. I guess your left. So what I just want to show is the Fox Foundation works on many different priorities, and all the way up here are field-wide challenges, so biomarkers. So, you know, when you go to a doctor and you have cholesterol levels that are changed or high, the doctor will say this is a risk factor for cardiac disease. We don't have a biomarker like that for Parkinson's. We don't have one that indicates that you may get Parkinson's. We don't have one that indicates or helps with diagnosis. We don't have one that helps with to see why some patients progress faster and some patients progress slower. So having an objective biomarker will be very important, whether it's in blood or spinal fluid, because right now the only diagnosis for Parkinson's is you going to a movement disorder specialist and doing clinical tests, and that can be variable. It's different in every patient. So I think our massive effort from the Fox Foundation is on this biomarker piece. Besides that, we do a lot of clinical trial recruitment, research tool development, and of course, public policy and advocacy in Washington, D.C. Moving to the middle is symptomatic treatments, and this is what you have now. You have symptoms. Can we develop drugs that make your symptoms better? And so, you know, you all take Cinemat, you all take different kinds of dopamine drugs, but we could have better dopamine drugs. You know, drugs that are better able to control your on-off episodes. And so we're funding work in this area for both motor symptoms and non-motor symptoms to look at, what are the new targets that can be utilized or leveraged for symptomatic treatments. What I'm going to talk about today is how do you slow progression? And I think that's not that the other pieces are not important, but this is the most important piece. With someone with Parkinson's, how do we, what are we doing to, one, understand the disease, two, identify new targets that can be made into therapeutics, and how can we set it up so that we learn from both the successes and potential failures of clinical trials in this space. So I'm going to talk about this. Not that this is not important, but just want to let you know that this is the focus of my talk. So very quickly, I think you all probably know this. Many of you live it on an everyday basis. Parkinson's disease, there's more than 6 million people worldwide. It's the second most common brain disease after Alzheimer's. And when you may hear me say it or the others say it, it's a neurodegenerative disease. There is a loss of dopamine neurons in your brain, and now it turns out that there are other types of neurons that are also dying in the brain, and that happens progressively over the term of the disease. And on top of that, you have what we call Lewy body pathology, which is this protein alpha-synuclein that I'll talk about in about 5 minutes, that also accumulates in the brains of Parkinson's patients. There's a paper that just came out early on this year from Boss Bloom. They did a global burden of disease study, and they showed that Parkinson's is the fastest growing neurological disorders. We expect already from 1990 to now, it's doubled, and we expect it to double up to 2040, and it may go up even higher. So what they call it as Parkinson's pandemic, but there's a real need and urgency now to do something about this disease. Okay, so what causes Parkinson's? And maybe I'll spend a little bit of time here just because I think it's important to set up, because I'm going to talk about a lot of genetic targets here. So we don't know. The simple answer and the most direct answer is we don't know why people get Parkinson's. We have some ideas, some correlations that say that certain things are correlated with increased risk of getting Parkinson's, but there is no cause or causation that's related to one single factor. So just to give you an example of the spectrum, it is a spectrum, and I think the genetic factors where if you have a particular mutation in your gene, in your DNA, and now we have recognized over 80 different genetic regions where if you have a variation in your DNA, it can increase your risk of getting Parkinson's disease. So those are, I would not say easy, but that's a thing that you can point to. It's a genetic reason why you may get Parkinson's. We also know that there are some environmental factors that have been shown to be correlated with the disease. So whether it's pesticide exposure or head trauma, things like that have been shown to correlate with increased risk of Parkinson's. I have to tell you, most of the people fall somewhere in between, and we don't know why they have Parkinson's. There are people who have exposure to pesticides and never get Parkinson's, and there are people who have never gotten exposed to pesticides and have gotten Parkinson's, right? So similarly with genetics, not everybody that has a genetic mutation gets Parkinson's. And I'll talk about this in a minute, but so the point is that there is a combination of a couple of things that are happening here. It's not just one single thing that you can point to. That even if you have that one single thing, that maybe there's something else that's happening in your body that then increases your risk of getting Parkinson's. So that's what we're trying to figure out. What is that predisposes people to getting Parkinson's? What are the couple of hits that need to happen? And what is that vulnerability that's inbuilt in your body that's driving the risk? So how do we identify new targets for Parkinson's? How do we go about finding out which target? A target is a protein in your body, most of the time. And we need to find these proteins because these proteins are what drug companies will use to develop drugs against. So how do we find them for Parkinson's? Well, the first obvious place to look at is the genetic mutations. So you'll see this is the biggest circle we have here, and this is where we start off, because we know that mutations in these genes are leading to changes in the protein, which then is predisposing you to getting Parkinson's. So that's the first place to start. But that's not the only place to start, because as I told you earlier, not everybody with a particular genetic mutation gets Parkinson's. Even if they have it, they may not get it. So we need to expand that circle out to other targets. And so that's what this is showing. So we have other pathways in our cells that we call mitochondrial dysfunction or protein handling pathway, oxidative stress, inflammation, I think you've all heard about. These are pathways in our cells. This is how our cells function. They talk to each other. They function themselves to do whatever their role is. And if we can find out what's the dysfunction in Parkinson's in these pathways, that may give us additional targets, protein targets to go after. Additionally, if we can now somehow relate what's happening in the genetic mutations and these pathways, so let's say there are mutations that then cause some change in inflammation, now you have connected this. So now, even if you have an inflammation drug, it would work on people that have the mutations, or if you have a drug that's targeting a particular genetics, that would work on a cause that may be happening due to inflammation. So I'll go into this in a second, but just want to give you a sense of that. The empty circles are things that we don't even know of. This is what we're constantly doing from your DNA all the way up to full body measurements. We're trying to understand what other pathways are affected, because there might be other pathways, other targets that we don't even know of. So what are genes? I talked a lot about genetics. So very quickly, genes are what's essentially in our DNA. And these genes are what makes us different, right? The different eye color, or even the risk of disease. So far, as I told you earlier, about 80 genetic regions have been linked to Parkinson's. 10 years ago, we only knew about 10. And in 1997, or pre-1997, which was before 20 years, we knew of 0 genetic links. So the more we find out, the better it will be. And I was talking to people yesterday that now we can sequence your entire genome for $1,000, and look at individual DNA differences. So the more we are able to look at what's happening, the more we can find out what may be driving you to have Parkinson's disease. And of course, these genes are what's in the DNA. Then the cell uses these genes to make proteins, and proteins is what cells are using to do many jobs like carry oxygen or aid in digestion. And as I told you earlier, rare variations in these genes are called mutations, and these mutations play a role in Parkinson's. So again, this is very important, so I just want to spend some time because I'm going to show you a lot of different genetic therapeutic targets. But before we go there, I think it's important to understand why we're focused so much on genetics. As I told you earlier, we don't know what causes Parkinson's. We know there are many causes or many things that are linked: genetics, pesticides, and then slew of unknown things that are leading to Parkinson's disease. So if, let's say, the change in your mutation, in your DNA is causing something to happen, which then leads to Parkinson's disease, maybe the pesticide exposure is also linked to that same pathway. So now, if you just have a genetic mutation, maybe it doesn't do anything. If you just had pesticide exposure, it doesn't do anything, but maybe if you have both, now they have some kind of effect together, which then leads you to get Parkinson's disease. So understanding these genetic mutations, that's what we know. We know what they are right now. It's very important to start to understand what pathways are being used, because maybe those same pathways are being used by many different things. That is how you get Parkinson's disease. And if we can understand those pathways, we can block them regardless of what the reason is, what the cause is. Does that make sense? So this is really important to understand, because this is how the field and therapeutic companies are approaching Parkinson's disease. They're starting at the genetics to try to understand the pathways and making genetic drugs against those genetic targets, with the hope that it doesn't just help people that have genetic Parkinson's, but helps people with Parkinson's at large. All right, and so I told you earlier, in 1997 was the first time we even knew that there was a gene mutation linked to Parkinson's. Before that was not the case. And that gene was synuclein, alpha-synuclein, and that's right up here. And this was found as a rare mutation in Greek and Italian families back in 1997. And at least for synuclein, if you have a mutation, what I mean by penetrance is there's a 100% chance that you will get Parkinson's if you have that mutation. These are very rare, extremely rare. There's probably maybe 100, 200, maybe 300 people in the world that have this. So this is not a very common event, but it shines a light on a very important protein. We also have this protein called LRRK2, LRRK2. That accounts for about 2% of PD cases. And again, I'm going to talk about these things. I just want to introduce them here. GBA1, which accounts for about 5 to 10% of PD cases. And then there is something called PINK1 and PARK mutations, which are linked to young onset Parkinson's disease. So these are, as I told you, there's about 80 gene regions that have been involved. There's some that we know exactly which genes are involved, but that still is about 10% of Parkinson's cases. About 90% of Parkinson's is, at least now, with the genetic regions we know, are not linked to genetics. They're happening because of some other reason, what we call idiopathic Parkinson's. Okay, so I'm going to jump into the different areas now, and hope that this is helpful to go into the different areas, because this is going to tell us all about the different therapeutic avenues that we're pursuing. So I talked about alpha-synuclein, and I think you should all know about alpha-synuclein for two reasons. One, I think I told you that in 1997, this was the first genetic mutation in Parkinson's that was discovered. In fact, before that, we didn't know, or we didn't think that Parkinson's had any genetic element to it. And amazingly enough, in that same year, we found out that, so this is the mutation in the gene. The gene makes the protein. In 1997 was also the first time that we found out that what's in these Lewy bodies that I talked about earlier, 90% of what's contained in that Lewy body is alpha-synuclein protein. So now you had, almost 21 years ago, a linkage to a target that's both from a genetic standpoint and a pathology standpoint was pointing to something very important here. So, by the way, we knew about Lewy bodies for over 100 years. Frederick Lewy, almost, I think it was in 1912, discovered these pathological protein aggregates in the brains of Parkinson's patients, and he called them Lewy bodies. He had no idea what was in those Lewy bodies. It was not until almost 100 years later that we found out that what's in these Lewy bodies is alpha-synuclein protein. So I think it's important to talk about what alpha-synuclein is because 99% of Parkinson's patients have alpha-synuclein pathology. In fact, that's sort of a post-mortem confirmation for Parkinson's is having this Lewy body pathology. So it's very important to understand this protein because drug companies are going after this protein in a major way. So I think some of you may have seen me do this last year, but I think it's important, so I'm going to do it again. So think of, because this is very difficult to understand, right? This diagram right here of a monomer and an oligomer, you may not kind of understand what that means. So let me kind of simplify this a little bit. So think of this as your normal alpha-synuclein, okay? By the way, alpha-synuclein is in everyone's bodies. It's people with Parkinson's, people without Parkinson's, we all have this protein. So let's say this is the normal shape of the protein. In Parkinson's, this protein starts to misfold in a way that we don't know what triggers that. Maybe it's that unknown cause, maybe it's pesticides, maybe it's genetics, maybe it's something else. But it starts to misfold. Now, and you know this answer from last year. So what happens if you put this through a printer? It's going to go in and come out on the other side, right? What's going to happen if you put something like this in the printer? It's going to jam. And then that's going to jam, and then what's going to happen to the things that are coming behind it? Those are going to jam. And there's going to be a huge ball of paper that's going to be in your printer. That's, in a very simplistic way, that's what's happening with alpha-synuclein. You have a monomer, a single molecule, and somehow what happens is it starts to misfold, it starts to go awry, and you start to get these types of molecules. And once you get these types of molecules, it starts to then fibrilize and then become a Lewy body, which is what's found in Parkinson's disease brains. So this is very important to understand. This is good. We want this. This is not good. We don't want this. And we want to remove this from our brains. So I hope that was important and understandable. Okay. All right. Perfect. So that will help in this. So what are the different types of therapeutics that we're going after for this protein? Now I told you this is a very important protein. All of you have it. There are many ways to go after this. So I'm just going to go one by one for all of these. So I told you what happens is you have your DNA. We all have it. It makes mRNA, and the mRNA that makes protein. This is the normal alpha-synuclein protein. This is this. It's the normal alpha-synuclein. This happens, this first three steps happen for all of us, regardless of whether we have Parkinson's or not. It's this normal synuclein becoming this Lewy body, which is what I showed you earlier, right? That's what's happening in Parkinson's. It doesn't happen with people without Parkinson's. So this is the critical step where the dysfunction is happening. And then, now we also know after all these years that some of this toxic protein is actually detected outside of the cells as well. So this is essentially your life cycle of alpha-synuclein. And we are leveraging this life cycle to make drugs. So the first way to make a drug against this target is right here, number one. You know what? If we had less of this, then there will be less of this happening, right? If we can reduce this so that this is not being misfolded all the time, then maybe we can reduce this Lewy body from forming or misfolding to happen. So that's what we call siRNA type approaches. These basically go into your DNA or RNA and try to lower the production of total synuclein or good synuclein. Because the idea is, let's reduce this so that there's just not there to make a Lewy body. A caveat here is that, you know, this is important. We all need monomeric synuclein in our body. So the balance here is to find out how much can we reduce this without having any side effects or safety issues. Number two way of going after this is right here. I told you this is a critical step where good becomes bad. right? If we can somehow make drugs that bind to the good synuclein and doesn't let it misfold, or binds to the misfolded synuclein and unmisfolds it, that's not a word, but I'm making it up, then that would be the best thing to happen, because that's going right after the bad target in your brain. So that's what we call, in sort of the industry terms, are called disaggregators. So they go after, either they reduce, prevent misfolding from happening, or they block aggregation or formation of Lewy bodies. The challenge here is that we don't know what this misfolded protein looks like. So how would you make drugs? How would you know where to go and bind? Because it's very difficult to know what this oligomer is. It's different in everybody's brains, right? So I'm giving you advantages and challenges in each of these steps. If you go to the next step, which is intracellular degradation, so all of our cells are built in with an internal garbage disposal. We all make proteins from our DNA. They do their thing, and then the cells degrade them. Why do they degrade them? Because we don't want those proteins. They've served their role, they've served their function. We need to degrade them because the DNA is constantly making new proteins. So if we can increase the trash compactor capacity of our cells, we can get rid of this, right? We can just get it out of the cell or get rid of it, and so that's what this is, which is the intracellular degradation. Can we increase our trash compactor capacity so that we just get rid of the bad protein that's misfolded, and we don't have to worry about it? What's the caveat there, or the challenge there? If we increase the trash compactor capacity for synuclein, there's like thousands of the genes proteins that our body and cell needs. How can we specifically do it so that it only increases the trash compactor capacity of our bad protein, but still keep the all the other good proteins there, right? So in each of these cases, there are challenges. And then the fourth way, which is the one that's furthest along in the clinic right now, is this number 4 and number 5, that if synuclein gets out of the cell, can we use an antibody or immunotherapy approach to basically mop it out? Can we just take a sponge and just sponge it out, just take so that they're no longer there in the brain? And this is the one that's really driving in the clinic right now. So these are the different clinical programs that are in the that people are testing in either people without Parkinson's or people with Parkinson's. I have to tell you, this is really exciting, because when I joined the foundation in 2010, there were 0 in the clinic. So in 8 or 9 years, there are now 8 or 9 programs that have advanced to the clinic. And just to give you a sense of this, what you're looking at, this is the company, this is the name of the drug. These are the different approaches that I just showed you. I showed you the 5 different approaches. This is the stage 1, 2, or 3, where they are. And then what role did MJFF play? And you can see in some of these cases, we did provide them funding. So about 4 to 5 out of the 8 or 9 programs, we funded them in the preclinical space to de-risk their programs and move them into the clinic. And what's really exciting is that the worry always has been that we all have synuclein, we all need the good synuclein. If you reduce synuclein, is that going to have any side effects or safety issues? At least until now, most of the trials that have happened in phase 1, there doesn't seem to be a safety signal. And so this is a good thing. That means that these drugs are safe. And Biogen is the one that's furthest ahead right now in testing their antibody therapy for synuclein. So I'll switch to LRRK2 and GBA since I have 10 minutes. That'll go a little bit faster now. So LRRK2 is the other protein, and again, I think LRRK2, just maybe a little bit more on LRRK2. LRRK2 is a kinase. It's a protein that activates other proteins. So, and we call them kinases. Kinases phosphorylate, and they kind of proteins talk to each other, and that's how they regulate each other. We know that about 2 to 3% of the population have LRRK2 mutations. So there are mutations, so it's important to understand. Mutations are at the gene level, And then what happens is when there are mutations, this LRT2 protein or the kinase activity gets increased. So if you have a mutation and your kinase activity gets increased, what the drug companies are doing is to reduce the kinase activity. And so right now there are, and let me talk about GBA and I'll talk about LRT2 programs in a second. The other genetic protein is called GBA1 or glucocerebrosidase. And you may ask what this is, and so I'm just going to real quickly show you a very quick slide on this. So look, here's the GBA1 gene, protein, excuse me. And what that does is it breaks down lipids or fats. So these glucosylceramides or glucosylsphingosine are fat molecules, lipid molecules that are important in our cells. This enzyme, GBA1, breaks down these lipids. And so, so if you think about it, if you have a mutation in this GBA1, then what's going to happen? There's going to be a reduction in the activity, right? Because it's mutated, it can't work. So this reduces. So if this reduces, and this is responsible for breaking this down and this down, but if this is no longer there, what's going to happen to these two? They're going to go up, right? Because there's nothing to break them down. And so this glucosylceramide and glucosylsphingosine increases, and that's what we think is pathological or leads to, for at least in relation to this gene, may lead to Parkinson's disease. So for therapeutic aspects, what we want to do is is to increase this or reduce that. And so that's what I'm showing you here. So you don't have to read all of this stuff here. This is a very simple way. I told you this is reduced, so we want to enhance GBA activity. And I told you this is increased, so we want to inhibit or reduce the accumulation of these lipids. So this is another way how we're going after GBA. So now to show you the different clinical programs. So again, here Denali is the first company that's in the clinic with a LRT2 kinase inhibitor. They've already tested it in phase 1 and have reported safety in their study. And there's many programs for GBA, including enhancers of GBA activity and things that reduce the lipids that are going forward. For example, Sanofi's Genzyme is currently in phase 2 with their compound. So let's talk about our favorite topic, repurposing. And sorry, I know this is going a little longer, but I think this is important to know, is to teach old dogs new tricks, right? So the idea for repurposing is it takes a long time to make a drug or bring it to a market. It takes 15 to 20 years. It can take over $1 billion. How about taking drugs that have already been approved for other diseases and repurpose them or rechannel them for Parkinson's? And that's essentially what this is, which is you have a drug that's approved for one disease, and how can we repurpose it for Parkinson's? And the famous examples for drug repurposing, I think you all know, is Viagra or Rogaine, both of which were not made for their final uses. They were being tested for other things, that there were some side effects which were apparently very useful. So, that's what we're trying to do in Parkinson's, right? So I'll talk about two drugs here that currently we're testing. One is GLP-1. So GLP-1 is a drug. GLP-1 is a target for drugs like exenatide. Exenatide is approved for type 2 diabetes. It's sold by brand names Byetta or I think it's Bydureon. And what does exenatide do? It stimulates insulin release from the pancreas by binding to the GLP-1 receptor. What we have seen is that there's an increased risk of PD seen in people with diabetes. Again, this is just a correlation, not a cause. It's possible that maybe Parkinson's and diabetes share same pathways. I talked to you about pathways earlier. Maybe there are similar pathways that they're both impacting upon. So if we have a diabetic drug, maybe that works in Parkinson's. And so this is the rationale for testing GLP-1 agonist in Parkinson's disease. Same thing with c-Abl. c-Abl is another target. This is a drug, c-Abl is a target for drug like nilotinib. Nilotinib is a chemotherapeutic. It's used for chemotherapy for CML, or chronic myelogenous leukemia. And its the brand name is Cigna. And again, c-Abl levels have been shown to be associated to be increased in Parkinson's. And c-Abl was shown in animal studies to reduce alpha-synuclein pathology, which I talked about earlier. So there's a good reason to go after this, this drug as well. And so here are some of the therapies that are being tested for GLP-1 and c-Abl currently. GLP-1, as you can see, there are many GLP-1 agonists that have been developed, and there's c-Abl inhibitors, and MJFF is working with Northwestern University to test nilotinib in phase 1 for Parkinson's patients. So maybe I'll just go through this very quickly. We are also leveraging, so I've shown you genetic targets, synuclein, LRRK2 I showed you repurposing examples, c-Abl and GLP-1. Now I'm going to talk about other therapies. So remember, this is what I showed you earlier. There are lots of other pathways. So why not leverage those pathways as well? There are drugs that just work on those pathways, like inflammation. How about testing those in the clinic? And so here again, lots of activity from different groups, testing either metal chelation, anti-inflammatory drugs, or mitochondrial enhancers, calcium channel blockers. Again, there are different pathways, and we need this. We need different shots on goal because we don't know what will work in Parkinson's. Maybe there will be 2 or 3 combinations. This is what happens in cancer. When you have chemotherapy, sometimes you get one drug, but most of the times you get put on 2 or 3 or 4 different drugs. Maybe that's what will be needed for Parkinson's. So the more targets we have and the more pathways we are testing, the better we'll be. So this is really the most sort of the summary slide, and I think something to be very proud about. Today's Parkinson's therapy pipeline is one of the most robust ever that I have seen, that we have seen in sort of the lifetime of the foundation. There are 41 disease-modifying therapies or things that are trying to slow the progression of the disease, and 30 symptomatic therapies. And is this enough? No. We want more and more of these therapies in the clinic. But I think this is a good, I think the place where we are at, where industry is engaged, where we have multiple targets to go after, and financial resources coming from foundation, from NIH, and from companies, I think is keeping this going forward. So what can you do today? We do need your help. I talked about biomarkers before. Every little thing that we can test in your body could be a potential biomarker. So I was talking about this yesterday. There's a group in Europe that's looking at, when you get cold and you get a goosebump, that is a pilomotor response that can be measured, and apparently it's different in Parkinson's disease versus people without Parkinson's. So that's a biomarker that we can potentially use. All of us wear watches or carry cell phones. These cell phones or these watches can actually measure our activity, daily activity, and maybe there's a particular motor activity pattern that could become a biomarker. We could measure in spinal fluid, we can measure in blood. There is a study where a particular breath of air could be a potential biomarker. People have reported there are differences in the way Parkinson's patients breathe, and maybe the breathing and the breath characteristics could be used as a biomarker. So every little thing from the DNA onto things like this, we are trying to go after as a biomarker. And if you're not able to be in a trial that is an interventional trial, a drug trial, a biomarker trial will also be extremely important because we want to learn so that we can use that as a biomarker in therapeutic trials. And if you can't be in any trials, you can do at least Fox Insight. Fox Insight is our online study where you contribute your daily data to us. And your data, your daily life is our data. So, this is an online study where you just go in and put in information on your tremors you may have, or if you've fallen down, or what have you. And you don't have to do this on a daily basis. You can do this monthly or however often you like. But all of that data then gets captured in a cloud. And there are data scientists that are actually looking at that data to look for patterns, to look for biomarkers, which would be very helpful. So if you don't know about Fox Insight, please go to foxinsight.org and be part of this study. And your caretakers can help you put in that information because they can see the symptoms that you're having that sometimes even you don't know of. And finally, you can join our community. I think what Donna and the Team Fox Detroit community is doing here. You could be part of something like that. You could join our monthly webinars, podcast, Ask EMD, where you can learn more and more about Parkinson's, and be part of Team Fox activities like the ones that are happening here. And finally, I know I'm about 5 minutes over, but we thank the Parkinson's community for their tremendous support and endless optimism. I talked about this earlier. Now I've come to know Mike and Donna and everybody else here in the community, and just the type of effort and dedication. And if you look at Donna's Facebook page and the amount of activities that happen every few months for Team Fox, without the help of people like you, we wouldn't be able to do our job. I mean, we have put in $900 million, but that money is important. It's raised by communities all around the world. And so, again, your optimism and the promise, and Donna said hope, is very helpful. And there's a good, I can kind of joke and say the state of the union is good in terms of the therapeutics that we have in the clinic right now. We want more, but I think your support and your tremendous optimism and hope is what will get us there. Thanks. All right, so I would like to introduce what we like to do here when we give a talk like this is, you heard from me what the Fox Foundation is funding, and that's great, but none of that is my research. I'm not doing any of the research. The research is, there are academic labs, there are industry and biotechnology companies that are getting funded to actually do the work. So while it was great to hear from me about, hey, this is where we put the money, or this is the strategy of how we're funding what we are funding, it's really important to know how that research money gets used. What's the type of research that actually happens? And what we like to do at these events is get a local researcher to come and speak to you about some of the work that they're doing. And I'm very happy to introduce, hang on one second, Leventhal, Dr. Dan Leventhal. He's MD, PhD, and Assistant Professor of Departments of Neurology and Biomedical Engineering at University of Michigan. He's a staff physician and Department of Neurology at the VA Medical Center, Ann Arbor. And I think his work is really just something that's, I don't think we even could think about 10 years back. So I talked about understanding the circuits and pathways before, but you can't do circuits and pathways in humans because it's not, it's not a very easy to access brain in live patients and live people. So we have to use really technological tools and animal models to do this type of work. Because we need to understand those circuits and those pathways so that we can have new targets besides alpha-synuclein and GBA and LRRK2. And this is a new concept. It's not very new, but it's something called optogenetics that Dan will describe. It's an unbelievable technology which you can leverage to really visualize the different circuits in a rat or a mouse brain. And something like this is what is going to be identifying targets of tomorrow, which hopefully will become one of the clinical trials that I posted. So I hope that you also find this an exciting talk. I'm really excited to hear from Dan. Optogenetics is so new and so defining, and I think it'll be good to hear from him how he's leveraging this technology for his research. So thank you all. Dan?
And if you have questions, feel free to raise those up and we can collect them. And if you have questions during Dan's presentation as well, you can make eye contact with anyone in the back and hold up your card and we'll come back and collect it so you can answer those questions.
Quick computer switch because I used a Mac and the videos don't show up in a PC. And also, I developed laryngitis 2 days ago and it was too late to find a replacement, so here we are. So thank you for the introduction, thank you for the invitation to talk and to Kuldip and Sarah, Lizzie, and Team Fox for letting me share with you some of the work that I've been doing at the University of Michigan. So as Kuldip said, I'm a clinician, so I see patients with Parkinson's disease in the clinic at the VA and run the deep brain stimulation program with one of my colleagues in neurosurgery. But I also run a basic science laboratory trying to understand what's going on in the brains of patients with Parkinson's disease. So Kuldip touched on a little bit of this. Every history of Parkinson's pretty much starts with that little essay on the left called An Essay on the Shaking Palsy. That was written by Dr. Parkinson, who's a general practitioner in England, early 19th century. He described 6 patients with what we now call Parkinson's disease. It was actually named Parkinson's disease. This guy is a famous dead French neurologist. Then Frederick Louis discovered Lewy bodies because all pathologists are humble and don't name anything after themselves. But then in the middle of the 20th century, the action really started going. And it was discovered that normal brains have these pigmented neurons at the base of a part of the brain called the midbrain, which is way down at the base of our brain. The Parkinson's patients don't have this. And it turns out that those neurons make dopamine, and that's the main neurochemical that's missing in Parkinson's patients. And we've known this now since the 1950s or so. Then all the new fancy stuff, the genetics, the cell biology, the functional imaging, the surgical treatments like deep brain stimulation came after that. But I'm going to go back and take a look at this. So the vast majority of treatments for Parkinson's disease in 2019 are still targeted at replacing that dopamine that's lost in the brain. And so we have Racinamet and a couple of other versions of Cinemet. We have synthetic drugs that look like dopamine to the brain, and those are things like Pramipexole or Mirapex. Vertigotine is the new ProPatch. There are things that boost levodopa level or dopamine levels in the brain. And generally, these will treat the slowness of movement and the stiffness. They usually treat the tremor, and sometimes we might even get away with treating the gait and posture problems in Parkinson's. But these medications have problems. So many of you are aware of dyskinesias, which I'll often describe to patients in the clinic as, ironically, the Michael J. Fox movements, because everyone knows what that is. But also people turning off, people turning on, we call motor fluctuations. Sometimes doses don't work. And then especially for the dopamine agonists, there's a whole class of problems called impulse control disorders, which have received a fair amount of popular press. Things like excessive gambling, excessive shopping, sort of obsessive compulsive type symptoms. So going back 2 slides, a little history lesson. So I teach a class on Parkinson's to undergraduates in the fall. And each week we read a different seminal paper on the history of Parkinson's research. And this year for the first time, or 2018 for the first time, I had the students read this paper. And this is sort of the paper that established levodopa as a treatment for Parkinson's disease. Here's the title. It was published in the New England Journal of Medicine, which is pretty much the most prestigious medical journal. And they started with 2 observations. One observation is that there's decreased pigment in the brains of Parkinson's patients. And the other observation is that there's decreased dopamine in the brains of Parkinson's patients. So I have these observations, and these really smart guys at the forefront of Parkinson's research did the obvious thing. They said, well, let's give these things back. So the first thing they tried is giving a hormone that stimulates pigment production. And what happened is absolutely nothing, except that possibly their skin tone changed. So then they said, well, let's give them something that gives dopamine back, and now they've got a winner. And the reason I tell this story is because this makes all the sense in the world at the time they did these experiments. And it turns out they were totally and completely wrong. But number 2 was a big success, and it's still a big success. So because the idea was you don't have enough of this, let's give it back, that sort of led to what I call the fuel tank model of dopamine. The idea here is that a Parkinson's patient, off of their levodopa, their dopamine tank is low, it's empty, and they move slowly, there's not a lot of facial expression. The dopamine tank is full because we've given the levodopa, now people start moving. That's a pretty simple, straightforward model. Some of you may have seen this video, it made the rounds. This is from the New England Journal in 2010. This is a gentleman from Holland who obviously has pretty severe Parkinson's disease. He's not moving very well, he's got quite a tremor, he's going to try to walk a couple steps in a minute. Now, without giving him any medicine, they take him downstairs, they put him on his bicycle, and this is what he does. The dopamine levels in this gentleman's brain have not changed, at least not a lot, since the last video you just saw. So that begs the question, do you really, really need the fuel tank to be full to move, or is there another way to get at it? And secondly, when the full tank is fuel tank is full, is it still possible to be moving slowly? There are a lot of potential reasons that this could happen, but I'm going to highlight one of them, and this idea really came from the addiction literature, because a lot of drugs of abuse work on dopamine, cocaine, amphetamine, things like that. And so I'm going to describe for you now sort of the idea for how this works. I am not saying that this is how the brain works, and I am not saying that I know what the dopamine levels are in Steph Curry's brain, but this is the idea. So you have something that's really hard, shooting 3-pointers. So how does your brain know that when you did it right, and you should repeat it the same way again? And the idea in the model is that every time that ball goes through the basket, or maybe for Steph Curry, every time he lets it go and he knows it's a good one, you get a little pulse of dopamine that tells your brain, that was sweet, do it again that way. Now let's say you're Nick Young, and you're pretty sure you hit it, and then you find out you didn't. So what's going on in Nick Young's brain? Well, I don't know. But the idea in the model, at least, is that you get a little dip in dopamine. It's sort of a disappointment signal. Like, man, you screwed that up. Try it a different way next time. The implication of this is that maybe the reason people with Parkinson's move slowly is not that you need the fuel tank to be full. It's that you can't get this little pulse of dopamine that tells you what you just did was the right thing to do and things are working well. And so you gradually wear off. And many of you probably have the experience that when you change your dose of levodopa up or down, things don't change immediately. It takes a week or 2 for things to level off. All right. So one of the things we're trying to address in my laboratory is really understand what does dopamine do in the brain, and specifically what does it do for the motor system. Okay? And what we're trying to do is test this fuel tank versus Steph Curry learning hypothesis. So can you move without dopamine, and might you move slow even if you have dopamine, and how do we refine that more carefully? So, but there are problems to studying this. So one problem is that, as Kuldip mentioned, it's really hard to monitor and control dopamine levels in the brains of living human beings. In fact, essentially impossible. We have some clever ways to do it with imaging, but they're not perfect. And you have to be sitting in a scanner while you do it, so it's not super practical. When we go to animal models, most animal models study really simple movements. You ask the mouse or the rat to run from one end to the other end of some sort of apparatus, or you ask them to push levers or poke their nose in a hole. I have yet to meet a Parkinson's patient that really wishes that their nose poking or lever pressing ability were much better when they were on dopamine. And then the ways that we have to make Parkinson's or to manipulate dopamine are pretty slow. So we can give poisons that kill dopamine neurons. And if you haven't heard of or read The Case of the Frozen Addicts by John Palfreman, who himself has Parkinson's disease now, I recommend it. It's the story of how dopamine toxins were discovered in San Francisco in the early 80s. But in any case, these things are slow, right? We need to turn dopamine off and on quickly, and we have to do it specifically. So these are the things that we want. We want a preclinical model of something that's hard to do, like shooting three-pointers, although I haven't been able to get the mice or the rats to shoot three-pointers. We want to be able to manipulate dopamine in their brains while they're alive very quickly and manipulate only dopamine and nothing else. And then we have to have some measurement of their behavior that's hopefully better than just lever pressing. So the first thing is we need a preclinical model. So this comes to us from a guy named Ian Wishaw, who's a researcher in Canada and has basically devoted his career to trying to understand movement through these preclinical models. So the point he's making in this video is that we can train rats and mice to reach for things. This is hard. This requires a lot of coordination across digits, right? This is not just going and banging the lever. And it looks a whole lot like what human beings do when they go to reach for little tiny things. And on top of that, if you give a rat or a mouse Parkinson's, they reach in much the same way that humans with Parkinson's disease reach. So we think we have a pretty good model of movement that we can study. The second thing we have to do is we have to be able to flip dopamine off and on. We have to be able to do it in a very precise way. So the brain is really, really complicated. Okay? Oh, that got loud. And it's sort of a jumbled mess of nerves and wires, right? So we draw these circuit diagrams, but that's not what the brain looks like. The brain is just a mess of wires all mixed together. So if the orange wires are the dopamine wires, how do we flip these things off and on and leave everything else untouched? And how do we do it with really, really, really precise timing? So a bunch of guys at Stanford invented this technology, give or take 10 years ago now, called optogenetics. And so what they invented is a way to use light to turn specific nerve cells off or on. So you turn the light on, the nerve cell fires. You turn the light off, it stops. Or you can do it the other way. You turn the light on, the nerve cell stops firing. And they invented some very clever genetic manipulations that allow you to do this in exactly the neurons that you want. So, for example, this picture comes from a colleague of mine at University of Michigan that also studies dopamine neurons. So this is a piece of mouse brain that has dopamine neurons in it, but it also has other neurons, other nerve cells in it. And we can label the red ones with this red marker, the green ones with this green marker. We know which are which. But if instead of using red and green markers, we use the proteins that allow the cell to respond to light, now we can shine light on these and only the red cells do stuff, and the green cells don't. Right? And the last thing we need to do is we have to be able to quantify these movements. So this clever group of computer scientists at Harvard invented this tool that they call Deep Lamp Cut. It actually sort of comes from the national, I don't know about the national security apparatus, but the sort of security apparatus. Because people were looking, how do you track people in surveillance videos? And so they trained computers to do this. So these guys at Harvard adapted those types of algorithms to tracking animal movement. So basically what we do is we tell the computer, we want to know where the digit's paws, we want to know where the digit's nose is, we give it examples, and it learns that. And then we can run our videos through it, and it tells us where everything is all the time. So when we do that, I'm going to show you just 2 data slides and not 2 deaths. So what we did is we did an experiment where we have our rats reach, and every time the rat reaches, we flip their dopamine neurons on. When it's not reaching, we don't do anything to their dopamine neurons. Down here, anything low on this graph is a normal reach. Anything high on this graph is abnormal. These two over here, those are before we do anything. This is the rat reaching normally. So then we turn the laser on, so we're turning on dopamine neurons only when the animal's reaching. And the key here is that on the first day, they're pretty normal, but not quite. And every day we do it, they get worse and worse and worse. Then when we turn the laser off, they get better and better and better until they're totally normal again. And what we conclude is that this really fits with that Steph Curry model. That it's not the fuel tank, because the dopamine levels here and here are the same, but the movements are very different. So then we did a second experiment, where once we had manipulated the rat's dopamine neurons, we turned the laser on sometimes and off sometimes, and on sometimes and off sometimes. And now what we find is that as soon as we turn the laser off, the reaches go back to normal. And as soon as we turn the dopamine neurons on again, the reaches get abnormal. Furthermore, it only happens if you do it while they're reaching. So if you do it when they're not reaching, nothing happens. They're totally fine. This, on the other hand, is very consistent with the fuel tank model, because now, when we change dopamine levels, the movement changes, and then it changes back very, very quickly. So what we conclude from this is that dopamine has something to do with learning motor memories. And we can then unlock those motor memories, maybe with or without changing dopamine levels. And they're stored somewhere, but we're not sure where. The question is, how do we access these in a meaningful way? Right now, the way we access them is by giving levodopa, or by giving Mirapex, or by doing deep brain stimulation. But can we be more clever about it, more specific? This brings me back to my earlier point. The brain is really complicated. So all of this stuff here, this is the super simplified model of the systems of the brain that are involved in Parkinson's. Okay? It's even worse than this. I mean, somehow we have to try and tease this all apart and figure out where these motor memories are stored and how are we going to get at them. But now we have the tools. So in the same way that we manipulated just dopamine neurons, right? Maybe we can manipulate this group of neurons, or this group of neurons, or this group of neurons, and we can step by step start picking apart how these things work and develop better deep brain stimulation. Or maybe there are other molecular targets that we can access. So in our lab, we're still working on these things. One question is, well, how do dopamine levels and experience interact? Because I showed you evidence that what really matters is what the dopamine level is when you're doing something. Well, maybe that's important for rehabilitation. Maybe rehab only works if you're on your levodopa. Or maybe when you do rehab, the way you do your rehab is later on the way you're going to access it. I don't know this for sure, so do not change your rehab practices based on this. This is speculative. All right, how does dopamine signaling normally work, and can we reproduce that? And so instead of just flooding the brain with dopamine, can we manipulate it on a finer scale? But the thing that I'm really kind of excited about is if we can understand the mechanisms in the brain that allow us to learn things in a manner that depends on dopamine, can we engineer that artificially? And more importantly, can we access those motor patterns that we know are still in there even when dopamine levels are low? And what are the targets that we might use to do that? So with that, I'm going to stop. Again, thank you for the opportunity to speak. These are folks in my lab that did all the hard work, especially Damien and Alex. My colleagues at the University of Michigan in neurosurgery work in the deep brain stimulation program and are a rather large group there. Thank you.
thanks everyone for handing in your questions. So Dan and I are going to go through some of your questions and try to answer them. One thing I would say is we're going to try to answer questions that are more general, that apply to everybody. So, you know, if you have more specific questions on your particular symptoms or disease, I think it would be more helpful to talk about sort of more generalized type of issues. So one of the questions that came is, what is the normal function of alpha-synuclein? So I told you that we all have it. Only 20 years ago we discovered that a mutation in alpha-synuclein is related to Parkinson's. Over the last 20 years, there have been many studies and investigators looking at this normal function. We think one of the things it may be doing is that it may be regulating dopamine release in neurons, which makes sense. You have a dopamine-related disease, and you have pathology that's driving that disease, alpha-synuclein pathology. So I think that makes sense. I don't think that's the only thing alpha-synuclein does, and there are studies currently ongoing. In fact, the Fox Foundation had launched an RFA last year to fund different ways to look at function of alpha-synuclein, normal alpha-synuclein. So we don't have the full answer yet. We're looking, there are some ideas, but right now we think it may be involved in neurotransmission or how neurons talk to each other.
Okay, here's a couple that are related, so I'll maybe address those at the same time. So there are two about relationship between smoking and Parkinson's. So there are very, very large, pretty well done epidemiologic studies, meaning take thousands and thousands of people, follow them for a long time and see what happens. That suggests that people that smoke get less Parkinson's, and also that people that use a lot of caffeine get less Parkinson's. This is a little bit controversial. People argue over, well, do people that smoke a lot die earlier from heart attacks and lung cancer before they get Parkinson's? And most of the research suggests that that's probably not it. What exactly it may be in smoking, whether smoking causes a decrease in Parkinson's, or whether people who are more prone to start smoking are less prone to get Parkinson's, or exactly how that works is not clear. People are trying to sort that out. I do not recommend starting smoking.
There's one that says, Fox Insight says, I talked about Fox Insight, the online study that you can all be part of. Question is, it says it's open to people with and without Parkinson's. What is the benefit of someone without Parkinson's to participate? How are we going to compare what's the quote-unquote dysfunction? We need people without Parkinson's as a comparison to what's happening in Parkinson's. This is tremendously important, not just for online studies like Fox Insight, but even clinical trials. And you know, you may ask, well, if I don't have Parkinson's, why should I do spinal taps, right? It's very important to have people without Parkinson's volunteer. This is one of the biggest drawbacks right now in clinical trials, is not getting enough people to be recruited. And having a profile of people that don't have Parkinson's is a good comparator or control to have to measure differences with Parkinson's.
So Kuldip just handed me one. Directly addressed to me, but unfortunately I'm not familiar with the small trial on muscle stimulation, but I'm happy to discuss it later. So I'll take another one. So another question, could a blood test show Lewy bodies and therefore confirm Parkinson's? So the short answer is no, and as Kuldip emphasized, that's really the question, is if we take lots of blood samples from lots of people that have Parkinson's, and as you just mentioned, lots of people Thank you. Lots of people that don't have Parkinson's, will we find something that's different in those blood samples? The answer to that is maybe, but we don't have a biomarker yet.
Just to add to that, this is the place where the foundation is putting a lot of resources. If we can measure alpha-synuclein, both the bad alpha-synuclein and the good alpha-synuclein, the mainly bad one, the oligomeric synuclein or aggregated synuclein or Lewy bodies, if we can measure them in blood or tears or saliva or urine, these are things that are easy to get from a patient, so you don't have to do a spinal tap. And if we can do that, we can use that as a biomarker. So it's tremendous effort being put into that, just to add to that. There's a question on thoughts on CBD oil to increase dopamine. So this is a topic that, frankly, I don't think we have time in the next 10 minutes, but I'll give you a very quick summary because this is a question that gets asked all the time about marijuana, about cannabis. So I think there are some difficulties in this research. One, if you get marijuana from 3 people on the streets of Detroit, there's going to be three very different formulations. Marijuana is not one compound. Marijuana is made out of 100 compounds. And what you all have heard about are the 2 main ones, CBD, which is the good marijuana, and THC, which is the bad marijuana. And so, so that's one issue. We don't know what combinations of CBD, THC you need. Do you need 20 to 1? Do you need 5 to 1? Do you need 4 to 1? And what you are getting off the street, you have no idea what's in there. 2, we don't know how much of that combination of CBD and THC you need. So those types of studies have not been done. So when you get things like, when you go to Amazon and order CBD oil, a lot of the times studies have shown that when people look at the chemical composition, sometimes there's not even any CBD in there. So, this concept of, using marijuana or CBD analogs for Parkinson's, it's a very difficult one. We don't really have quality material. It's not standardized. People are using different things. Not everybody's scientifically savvy, so if you're getting something from Amazon, you're not going to check. Let me look in the back and see what the proportion of the 1,500 chemicals are, right? So it's a very difficult thing to study and address. This is something where, a study like Fox Insight can help with, because there are people doing this, and we can ask them what they're taking and what kind of symptoms are being helped, what kind of symptoms are not being helped. And the really scary part is that marijuana is a drug. We have no idea about side effects, right? So everybody talks about, oh, it didn't work for this, or it didn't work for that, but did you have side effects of using marijuana? So, or CBD. So I think it's a topic that's very difficult. We also don't understand how marijuana works, what kind of receptor targets or protein targets it works through. Is it really through CD1, CD2 receptors? There's a lot of research still to be done, and this is not easy. Even a questionnaire is not going to help because, as you know, it's not legal in all states. So people may not be very open to sharing their experiences in terms of what they've used. So I would say consult with your physician before you would do anything like that.
So, I'm being contrary to just add one thing to that. So I completely agree. Every study that I'm aware of marijuana or marijuana derivative in Parkinson's is a very tiny study, and they've had very contradictory results. And all of the things that Kuldip just said apply. We don't know what's in all these preparations. However, I will argue that we don't really understand how levodopa works either, and it works. So, yeah, is it plausible that CBD could be helpful? I mean, There are receptors for CBD all over the parts of the brain that are important in Parkinson's. But the bottom line is, we don't know yet. There's lots of stuff in the brain that might be useful that we don't know about. And I think CBD is well right on that list. So this is sort of a general question I want to address. What is the difference between Parkinson's syndrome, Parkinsonism, and Parkinson's disease? So, when I tell patients, when I first see them and they look to me like someone that has Parkinson's, I tell them that they have Parkinsonism. And Parkinsonism is an adjective. It's like saying that you have red hair or you're tall. Parkinsonism is just an adjective that means you look like someone that has Parkinson's. There are lots of things that cause Parkinsonism. So the obvious is Parkinson's disease, which is really defined by Lewy bodies pathologically in response to levodopa clinically. But there are certain drugs, especially certain drugs that used to treat nausea and drugs used to treat things like schizophrenia that can cause Parkinsonism. And there are other degenerative diseases that look like Parkinson's but are not Parkinson's disease. So Parkinsonism is just the descriptor that says sort of tremor, slow, rigid. Parkinson's disease is the pathologic process with Lewy bodies that responds to levodopa.
Okay, so a little bit related to this interesting question, if all PD patients get Lewy body deposits, then why not most Lewy body disease? Most have Lewy body disease. So, and how are those patients different than PD? And so maybe Dan can speak about this from a clinical side. Pathologically, this is what I think Dan was just saying about Parkinsonism, that we don't think it's one specific disease, that it's a spectrum. And some have pathology that correlates with clinical symptoms, and some have clinical symptoms that are, may have cognition as a main issue, but the cognition may not be an issue until 10 years into the disease. So I think, they may be on the spectrum, and what we're trying to do from a pathology standpoint is trying to understand, that people that have clinically been diagnosed with LBD or DLD, do they have a different, synovial profile than people with Parkinson's? People are trying to understand if the aggregate that's formed, the Lewy body type aggregate that's formed, if that's different. Is it in a different cell type? Is it in a different tissue? Is it in a different region of the brain? So I think we need to understand more of the spectrum of diseases. The interesting thing is that they're both synucleinopathies, synuclein aggregates in both of them. So if we can have a synuclein therapy, it could work potentially for all of those synucleinopathies.
Yeah, so just to add on that from the clinical side, one way to think about it is exactly what Kuldip said, that someone who has had Parkinson's disease for 15 and 20 years and then develops the dementia syndrome that we associate with Lewy body dementia, which is, I believe, the diagnosis ultimately given to Robin Williams, the pathology looks very, very similar. But if it starts with sort of the Parkinsonism and the dementia at the same time, we call it Lewy body disease. I want to answer this one. I actually don't know the answer to this question, but it opens up an interesting discussion. So the question is, what about a virtual reality mask, gaming, increasing reaction times and balance? So the short answer is I am unaware of studies looking at virtual reality in Parkinson's, although I am certain that someone has done it by now. The slightly longer answer is that people are really asking about what can we do to slow the progression of Parkinson's disease. And the one thing that always shows a signal is exercise. And whether that's actually changing the brain itself, or it's just you stay strong and it makes you more resilient, or whatever it is. But almost all the studies are modest exercise, 20 to 30 minutes, 3 days a week. Whether it's a brisk walk, stationary bike, there's boxing for Parkinson's, dancing for Parkinson's, Tai Chi for Parkinson's. All of these things seem to do something beneficial. And what I always tell my patients is the right exercise is the one that's safe and the one that you keep doing.
So there's a question on status of stem cell research. Again, this is something that we could talk for hours. So again, the idea, you remember Dan's slides, dopamine is gone, and the idea, you could either increase dopamine by giving levodopa, or you could give stem cells into the area of the brain where dopamine is gone, so that you have more dopamine neurons making dopamine. So this is obviously not a new area. This stem cell trials happened in the 90s. And some of the things we learned in those trials, it was in a small number of people, but there were dyskinesias that were precipitated when some of those patients that finally passed away, when we looked at their brains, the newly grafted tissue actually had Lewy body pathology. So now, you put good tissue or good cells in a quote-unquote diseased environment, and now that the good tissue is basically getting Lewy body pathology as well. So, there are a lot of caveats to stem cell research. All the other issues that come with that, around, you can't do brain surgeries, in 8 different places where neurodegeneration is happening. It's not like there's only one area in the brain where this is happening. And the disease progresses, and there are other areas of the brain and other cell types that are dying. So there's no way to control something like that unless you are giving stem cells peripherally as an injection. And if you're giving it as an injection, then the caveat is, how do they get into the brain? And how do they get into the brain and know how to get to the deep areas that Dan was talking about earlier in the midbrain? How do you know that if you're giving them as an injection, they won't stay out in the periphery and actually have other side effects or may induce tumors, right? So there are a lot of challenges to stem cell research. That doesn't mean it's not happening. People are trying to find out ways to overcome the blood-brain barriers so that it can get to the brain. They're trying to come up with stem cells that can be more targeted. But I always get worried when I drive sometimes in my local roads in New Jersey, and I'm listening to Saturday mornings, and there's an ad that comes on for a local doctor in New York who is injecting stem cells. Your own stem cells are being injected for every disease that you can think of. To me, it's very scary because really there's no research right now that shows that a particular stem cell given a particular way behaves in a particular way, right? So I think it's important research. We're using stem cells from Parkinson's patients in a dish, what we call iPSCs, so that we can study them and see what's different in patient stem cells. That's I think has been the most progress that we've done in the last 10 years with patient stem cells, is looking at mechanisms of what may be different in Parkinson's versus people without Parkinson's.
This one's been sitting here for a while, so we should address it. So the question is, my mother and brother both had Alzheimer's. Is there any relationship with Alzheimer's and my Parkinson's? So the short answer to the specific question is probably not. There is some relationship between the neurodegenerative diseases, just sort of related to another question here about, are the plaques the same in Alzheimer's and Parkinson's? And they are not the same, but they do have some features that overlap, and there is some question about whether Alzheimer's and Parkinson's and other neurodegenerative pathology can sort of feed on each other. But the reason I like this question is it brings up the family history question. So there is, people will commonly ask, what's the risk of my brother, uncle, kids having Parkinson's? And the answer especially for sort of older age onset Parkinson's disease is essentially the same as my risk of getting Parkinson's. Maybe that much higher, just a little tiny bit. And that's in general true unless you carry a Parkinson's gene, whether young onset, old onset, whatever. If you don't have a strong family history, the risk to family members is essentially the same as my risk. And in fact, even in families where there is a history of Parkinson's, say multiple siblings, Parkinson's is common. It happens in 1 to 2% of the population over 60. So for a pair of siblings, that's about a 1 in 1,000 chance that any two siblings just by chance might have Parkinson's, which isn't super big, but it's not zero. I mean, it happens with a fair degree of frequency. So the short answer to the question is probably not, and the risk to close family members, unless you know you have a really, really strong family history, or you have an identified gene, is it's essentially the same as everybody else's.
Dan, do you want to take this one? Sure.
Yeah, so why do PD symptoms vary so greatly? Okay, so again, the short answer is I don't know. The long answer is that there are lots of different causes of Parkinson's. We know that now. So, as Kuldip said, when I was in medical school in the early 90s, Parkinson's, mid-90s, Parkinson's was one disease. Beginning in 1997, genes started getting identified. We now have over 80 genes, so right there's 80 different Parkinson's. Plus, maybe Parkinson's from head trauma versus Parkinson's from pesticide exposure versus Parkinson's because you injected a bad batch of opioids in 1982 in San Francisco. And so because there are all these different causes, that's probably why the symptoms vary so much. And even if for people with the same cause of Parkinson's, neurodegenerative diseases is like real estate, location, location, location. So you can have the same pathologic process in two different parts of the brain that control two different functions, and you will get two different manifestations. And so if you get Lewy bodies in one set of cells of dopamine neurons, maybe you get more tremor. If you have it in a different set of cells, maybe you get more slowness. So it's not really clear other than to sort of wave our hands and say Parkinson's is not one disease.
Okay, we're coming to our final question. Sarah is waving me from the back. So I'll take this opportunity. There's a question here to kind of plug something for the Fox Foundation. So the question is, what is the drug they're talking about in Ireland? Is it promising? Should we move? So I don't know which drug this is. We're talking about in Ireland. I think I showed you, the therapeutic landscape today, and I hope you appreciated that there's a lot going on. But more needs to be done. About moving, I mean, as you know, just because you have Parkinson's and you move to an area where the trial is happening doesn't mean you're going to be necessarily part of the trial. But if you're so inclined to be part of a trial, and as I said earlier, we need you to participate in these trials, please go to foxtrialfinder. I think it's over. And the Fox Trial Finder, if you don't know, is a match.com type of service. So you say you live in Ann Arbor, you have this many years of Parkinson's, these are your symptoms, and you're interested in being part of a research trial in 15-mile radius, let's say. Let's say there's a trial that opens up, and they match, you match up with what they're looking for, that you get a email letting you know that you're matched, and if you're interested, the trial will get in touch with you. Now, again, that doesn't mean that you can't say no at that point. It doesn't mean that you won't get selected. But again, it helps for drug companies to have a ready volunteers of people who are ready to get into the trials. So, both people with Parkinson's and without Parkinson's, if you could go and to Fox Trial Finder, I know I'm on it as a control, and it would be very helpful. So I'm going to wrap up, and first of all, say thank you to Dan for coming out today on. Today on Saturday and spending time and providing information to all of you. I want to thank all Saturday and sitting with us and hopefully learning some of the really interesting things that are going on, both from a funding standpoint, but also, as Dan showed, from a breakthrough research standpoint. Again, to Donna and the Team Fox Detroit and everybody in this community who is helping us, your hope, your optimism, your faith in us is what keeps us going. Thank you all for coming. Please leave your feedback cards at the table or provide them up front if you can.